I'd like to contribute to this session of central dogma, which emphasizes the role of RNA between protein and the DNA. As we know, this is a different version of a central dogma emphasizing the DNA as the storage of genetic information, and mRNA produced from DNA to produce a messenger to instruct the protein synthesis during the process of translation. But however, we now know RNA is more than messengers. As illustrated here, there are so many different kinds of RNA. When simply mRNA as a messenger, rRNA actually is a part of the protein RNA machines to make a translation to happen, and the tRNA is a part of the that machine too to transfer RNA, amino acid to the ribosome. And we know there was a huge population of heterogeneous RNA in the nucleus, and many non-coding RNA, both in the nucleus as well as the cytosol, as well as many other regulatory RNA, not defined RNA modifications, some other functions, and a bunch of regulatory RNA, big or small. Okay, so this is constitute a very, very busy world of RNA world to contribute to the diverse function and the regulation in the cells. So this actually created a new frontier, so-called the regulatory RNA, the function of RNA in regulatory biology. Illustrated here is summarized mechanistic insight into how RNA function as a regulator in mammalian cells, that including RNA serving as a scaffold to bring many protein together. RNA function as a decoy to prevent or regulate the interaction of protein with other protein or DNA, or RNA as a guide to guide our other machinery into a specific location in the DNA, typically exemplified by CRISPR system, the machineries, and also RNA as a signal for integrator or activation of different complexes. And in fact, if you take a look at the RNA function inside the cell, and you can actually one-to-one to actually point out the similar function. Oh, I'm getting ahead of myself. Similar function of RNA that has a catalytic function like a protein. RNA can serve as a structural components like a protein. RNA can mediate protein-protein interactions, and RNA can serve as a chaperones, molecular targetings, and so far I could not find any evidence that RNA get conjugated with lipid, but might happen in an indirect way, and many different type of RNA modifications. So in this regard, we can say that RNA may have many functions as protein do, but just divided labor in different contexts or in different biological process. So in this regard, I'd like to show you this picture that happened 30 years apart. In fact, this is how the time I joined the laboratory of Tom Mariatis, because this paper published Tom and Robin Reed, postdoctoral fellow, then about this isolated spisosome visualized and the EM, known or jokingly as a popcorn structure, because at that time we know nothing about any of those components in the spisosome. Yet 30 years later, when the whole structure get resolved by my colleague now, Yigong Xi, and published in Cell, this is exactly 30 years later when every component is known, and you can see how amazingly the structure are related to each other. As if we have a high-resolution microscopy 30 years ago, we don't need to go through such a labor to purify each component and characterize their functions. In fact, this make me wonder whether or not there is a reverse alpha-2 that we can just get a structure first before we know the protein, and then we can deduce their machines. maybe those things can happen very fast. And it is these structures that guided me to the field, and not because at that time we know nothing about the protein components. We start actually take an approach to inject the whole thing into the mice and make a monoclonal antibody and characterize one by one to see which one is the real factors, how do they contribute to the splicing pathway. That's a long stories. And one of the findings, unexpected finding, is that when I get my first antibody, my lab mate and a roommate, in fact, in Tom's lab, was a Xiangdong role, happened in our room. Each room has just 2 people, and a microscope was sitting in our room. And he said, why did you take a look? And the microscope, what's look like with the splicing factors. And it turns out this gives us something we stunned the first time when we see under the microscope. It's a beautiful speckled structure. And this structure actually has guided me for in fact, so many years afterward as this molecular condensate. Sunny gave a first talk in this session about his view about the molecular condensate. We can continue to debate what that means, because actually that reflects the current status of the field and what's going on here. And maybe we can debate continuously. One of the view, at least you can consider alternative view, is that this kind of spectrus maybe is the DNA scaffold with proteins, scaffold for RNA that coordinated many coupling process during transcription. You may call them molecular condensate, you may call them a transcriptional hub, or some kind of a special domain or nuclear subdomain that exists in nature, exists in eukaryotic cells. Okay? So then that brings me the questions, what kind of a regulatory RNA involved? Because if you inhibit the transcription or get rid of RNA, the whole structure gets changed or collapse. And in fact, one of the type of RNA is what we later discovered as so-called eRNA or enhancer RNA. And turns out the enhancer is not just a platform for protein to bind, but rather also produce RNA, just like a promoter, but in an asymmetric way. Illustrated here is androgen receptor regulated genes, and there's an enhancer upstream of these genes that bind to androgen receptor. And if you do this detect of nascent RNA, you can see the transcription getting induced, and then this produces RNA in this symmetric manners in both forward and backward strand. But unlike gene promoter that's produce RNA in one side and a small RNA in the other side, but very short, this one is equal length. That's so-called eRNA. And because we know this RNA play intimate role in chromatin biology, and we actually wondered how many RNA there to function. In fact, so we developed this technology called GRID-seq. This is a typo, sorry. There's a global DNA RNA interaction with DNA. It goes like this. DNA, hypothetically with a protein RNA complex, then we truncate down with restriction enzyme, and then add this linker with a biotin so that one end can only ligate to RNA, the other one ligate to DNA. Then we can isolate this fragment and go through sequencing. And you can see most of the RNA remain associated with a template at the site of transcription. But most of the time, you can detect their interaction with distal DNA element. In fact, in most of the cases, those elements corresponding to enhancer, well-known enhancer. This actually allows us to deduce a global picture of how promoter interact with enhancer in every single cell types. So we have not yet extended this technology to single cell yet. I heard Sandy's lab is actually doing make that effort. So with the emphasis of RNA, this is a historical review of our journey in research RNA. And today I'd like to bring all the way to the up front using this PVT1 as a paradigm. It's a lncRNA gene. Okay, it's a long coding RNA gene. The reason is the interesting paradigm is that this non-coding RNA gene is known as an oncogene, but at the RNA level, but also known as a tumor suppressor, but at the DNA level, how this happens. So PVT1 RNA is oncogenic has many evidence. For example, is actually sitting right upstream of the promoter. This is a famous oncogene, and the transcript has been shown to stabilize and make proteins. But the transcript can do many other things, including multiple microRNA, including interaction with DNA, regulate the cell cycle, contribute to drug resistant, and contribute to the single activation pathway. So there is a wealthy literature document the oncogenic role of this non-coding RNA. What's interesting was the discovery a few years back by Howard Chen lab, and showing that the promoter of this PVT1 gene actually functions as an insulator, not to prevent the enhancer within the gene body of PVT1 to interact with MYC. Okay, and then so therefore the DNA is a tumor suppressor. When you get rid of it, it actually suppresses enhanced tumorigenesis. Okay, how this paradigm get utilized in the cell to promote genesis become a fascinating questions. Okay, so now let me introduce ecDNA and illustrate to you how this ecDNA might be explored, this kind of a paradigm to tilt the function toward oncogenesis over the tumor suppressors. What's the ecDNA? Turns out we have a true expert of ecDNA here, Don Cleveland, my former chair, and he will tell you probably some of those stories. ecDNA represents extrachromosomal DNA that actually spill out from the regular chromosome as a result of a genome instability. Okay, and then they become a circle. So we're not talking about the circular RNA. In fact, Hong Wu will tell you PVT1 may have a capacity to make a circular RNA, but we're talking about a circular DNA at this point. Okay, and when you have such a, again, this is Howard Chan's paper, when you have this ecDNA, this is from the prostate cancer cell, colon cancer cells, GBM brain tumors, and also gastric tumors. And you can see this ecDNA carry this MYC locus, some others carry this EGFR locus, and they form clumps together. They call them ecDNA clumps, speckles. And they show in this paper that ecDNA can actually, each different type of ecDNA can form different type of clusters. This is radiate, this spots between the distance between two spots. This is autocorrelation. So this line means they form really tight clusters, and this is loose clusters, and then this is random distributions. Okay, indicating that the ecDNA has a different tendency to interact with one another, and they show by that mechanisms they can enhance gene expression through trans mechanisms. Okay, and how may ecDNA amplified oncogenic activity while mitigate tumor suppressor function to promote tumorigenesis is the questions we like to address in these studies. Okay, and then this is the slides from Adrian Ren and another author, I forgot, lab in publishing cancer cells, and they performed the Hi-C experiment. I did not go, I think I don't need to go to detail, and with this chair pad to detect a protein-protein interaction. But in this particular cell lines, it harbors a ecDNA, big ecDNA. What they found out is that the DNA, normally you have this diagonal line indicating the local interaction, and they found this DNA can interact practically most of the DNA in other chromosomes, so-called trans interactions. And the same thing happening here. But if you carefully think about this, this could mean one of the two scenarios. One is that this is your ecDNA, they just go out to visit different neighbors. Then you get this kind of multiple interactions. Or the other way around, multiple neighbors were folded back to visit you in your house. But how can we distinguish which is which? We don't know in this case. Therefore, we utilized a control in these studies by actually compare ecDNA with HSR, standing for homogeneous staining region. By fish, you can see they are locally amplified, but ecDNA, they are scattered around in these fish studies. And in this case, we performed similar Hi-C, and as you can see here, that with the ecDNA, with SHR, they basically produce the same thing. Okay, so as a result, but however, we believe, as we will see, that actually this SHR representing all the genes loop to this locus to interact, or ecDNA is the other around. And the evidence comes from DNA protein interaction mapping I just mentioned earlier. In this case, you can see here, this ecDNA, you have this line of, this is RNA, this is a DNA, this is RNA interact with this is RNA from MYC locus, there's a PVT1 locus interact with every piece of DNA. And conversely, this DNA interact with pretty much every RNA in this locus. Okay, but however, when you have this local amplification, HSR, MYC, then you see this RNA interact with the locus, the DNA interact with RNA, but not RNA with the DNA. So in the other word, the interpretation, So in the other word, this is DNA interact with a lot of RNA. RNA just come over to visit the same locus, but a lot of the other way around. So this distinguish the ecDNA with the local amplifications. Okay, so what's the role for this RNA? And it turns out if you use ASO to destroy this PVT1 RNA, as quantified here, and we did a Hi-C, GRID-seq, ATAC-seq, all those kind of a seq. And you can see here, this is a control, there's no difference. Without ASO treatment, this is SHR, but with ecDNA, you can see the lung target has no difference, but the target, they start to interact less based on this measurement of so-called trans-chromosomal interactions. So it's a complex formula to calculate such things. So at the cellular level, you can also see by fish that upon the disruption of the RNA, it started from the speckles and then become dispersed as a quantified this. So put together, this give rise to this idea that how ecDNA explore this kind of a new configuration to favor tumorigenesis while mitigate its role as a tumor suppressor. This is done by forming this circle so that this PVT1 function is insulated to MYC. But however, once we liberated from the local context, it starts to engage trans interaction so that its enhancer can, using trans mechanism, to either join the existing hub to interact the cellular genes or form their own hub to enhance each other's expression as a result to enhance mutagenesis. At least this is a hypothesis we can think about, and the following experiment will test that models. So I'll stop here, and thank you very much.